good afternoon. My name is Sandro Zampieri. I'm the director of the Department of Information Engineering. And it's a great pleasure to welcome you all today on behalf of the department that today is offering the third and the last distinguished lecture of the series that we are proposing this year. After the first lecture devoted to machine learning, the second devoted to quantum information, this third lecture is about neuroscience. So three completely different subjects. And the reason of the choice of neuroscience is because we think that this discipline is a great opportunity for our department. We can offer a lot of tools and know-how, like in signal processing, in imaging, in modeling, that could be of great help in the development of neuroscience. So I think that we have already a very good group working in neuroscience here, and Alessandro Bertoldo is here and is one of the main representatives of this group. But I think we can give more, and through also collaborations with other departments and also with Maurizio that is here and is our colleague in this university, I think we need to propose our know-how for the development of this discipline, because the impact of this discipline will have in the future will be very important for us, for our development. And so we cannot leave this opportunity. And so for this reason today we are proposing this subject. I hope you will enjoy the lecture. And now I pass the mic to Grison, my colleague, that will briefly introduce our guest. Thank you.
Okay, good morning, everybody. So let me introduce a little bit of Professor Corbetta that is here, that from this year is chair of neurology and professor of neurology at the Department of Neuroscience of our university. And is presently still, I guess, NOMA's top professor of neurology at the Washington University School of Medicine in St. Louis. He received many, many awards in the last 20 or 15 years. From McDonald Foundation, Cognitive Science, Marie Curie Chair from the European Union, the Academy of Neurology. He has Distinguished Lecturer and Senior Investigator Award from Washington University. And most notably, from 2010, he entered the top 100 Italian scientists. And he published a lot of papers, of course, and he was nominated a highly cited researcher from Thomson Reuters in 2012 for the previous decade. That is an honor shared only with other 130 more or less investigators worldwide in the field of cognitive neuroscience. Professor Corbetta is internationally known for a multidisciplinary science and research on the neural basis of human cognition and the neurological mechanisms underlying stroke recovery. His research focuses on how physical organization of the brain relates to behavioral recovery after stroke. His work on stroke-related attention deficit disorders has provided some of the first direct evidence that brain injury can cause functional changes in brain regions far from the site of the actual injury, which are related to behavioral impairment and recovery of function. For attaining this, he works in a multidisciplinary research area where doctors, psychologists, biologists, physicists, mathematicians, and engineers share their expertise to advance our understanding of the brain. So it's our great pleasure to have him here, and let's welcome Professor Corbetta.
Thank you so much. So it's a great pleasure being here. Can you hear okay? Po forte. So I've been giving a few lectures over the last maybe couple of years in Padua. And so I'm kind of scraping the barrel here. Okay. So a few months ago, we had a day here at Day in which we discussed brain networks and the whole idea of studying brain networks using imaging. I gave a few lectures on the history of neuroscience and so forth. So I was a little embarrassed here today. What am I going to talk about again? So I decided to give you, I guess, a story that I've been working on a lot for the last 5 years. So it is a bit narrow, perhaps, but it deals with a very important problem. And I guess you can get a sense on how much you can get done in 5 years, basically, by working carefully and in a disciplined way on a particular topic. And this is a topic that, of course, is of interest to psychologists, to neurologists, but it's also of interest to engineers and physicists, because we study this topic, which is the effect of stroke on brain networks, not just for understanding stroke, but also for understanding the organization of brain networks and how networks change depending on physical alterations of the matrix of structural and functional connections. So I think there are theoretical issues here, there are practical issues, there are very clinical application issues as well. So it's a little different talk than, say, what are the biomarkers of Alzheimer's disease, or what are the biomarkers of multiple sclerosis. I think we're studying this because we're interested in basic neuroscience. All right, so before I start, I'm going to set up kind of what we know, what I carry in my pocket as I see patients, what I teach residents, in terms of the effects of stroke on behavior, okay? So everybody knows that a stroke is basically a drop in blood flow. There is a clot, there is an obstruction, one of the blood vessels in the brain is obstructed, and so there is a decrease in perfusion that leads to a decrease of oxygen and a decrease of glucose. And within a few minutes, 4, 5, 10 minutes, that area of the brain will die. And this will lead to local dysfunction of neurons, glia, and connections in and out of that region. Hence, we're going to have different neurological deficits depending on where is the lesion in the brain. So one of the principles, one of the cornerstones of neurology and general cognitive neuroscience is that different areas in the brain are responsible for different functions. And depending on where you hit the brain, you're going to have different deficits, okay? This is also called the place theory of brain function. So as an example, I'm just going to show you a patient that is striking. I've shown a few times, so I apologize for those of you that have seen it already. So for instance, if you have a lesion in the left side of the brain, on the frontal lobe here, just in front of your ear, and now you have, that is an area that is being responsible for speaking, you will have a deficit of speech production. People will have trouble speaking, but they will understand, or they will be able to read. And the other thing that people believe, and this is what you teach residents, is that if you have now two deficits, say you have a speech problem and also your right arm doesn't work very well, that means two neighboring cortical modules in the cortex are affected by the lesion. So if the stroke is affecting a big blood vessel, then a lot of the brain is going to be suffering. Therefore, you're going to have several lesions, okay? So let me give you an example of this idea of selective deficits, okay? Which is kind of the straw man, the odd man out that I'm going to set up against this network view of brain function that I'm going to discuss in the rest of my talk. So this idea, rarely in science there is kind of a moment, you know, Einstein writing his paper back in the patent office in Zurich. In neuroscience, the moment is 1861, where this gentleman is a surgeon, he's not even a neurologist. He basically made this observation that a patient with a very selective lesion of language was having a deficit, a lesion in a very specific part of the brain. Up to then, there was this idea that the brain was kind of not very specific, not very organized, so that the more you hit the brain, the more deficit you have, kind of a principle of mass action. But this observation showed that you have a lesion in the brain, you have a very selective deficit. And the story is kind of an interesting story. Broca was a surgeon. He was also the president of the Society of Anthropology in Paris. And throughout the winter of 1861, they were discussing localization of function. And so when this patient, Leborn, came by, and he was a gentleman that developed gangrene and abdominal dysfunction, he had some kind of an acute abdomen, he was referred to Broca. But Broca, mindful of of this dysfunction about localization of function, realized that this patient could only say one utterance. He could only say ton, ton, ton. That was the only thing that he could say. But yet he could understand everything. And so when the patient died of abdominal complication, Broca, with some of his bodies from the Society of Anthropology, did an autopsy. And they basically show for the first time that there was this lesion in the left frontal lobe that was very selective, and the patient was having this very selective deficit. And the brain, they realized this was a special brain. In fact, in contrast to what was usually happening, or is still happening, there was no so-called brain cutting. The brain was never cut, because they realized in that moment, in April 1861, that this was a seminal observation from neuroscience. And if you go to Paris and if you pull a few strings with the dean of Paris 5, you will go and there is this museum, the Museum Dupuytren, in which there is this vat, and there is still the brain there. You can go and see it. It's really quite impressive. So anyway, I'm going to show you the patient, pretty much the same patient that Broca saw in 1861. And the whole point here is that the deficit is very selective. Okay?
And tell me, why is it that you're moving, actually? All your children are grown, right? How many children do you have? What number is this? Right? Right. Now let's count it out.
1, 2, 3, 4.
Nice, nice. Well, since you did 4 so well, let's go for broke. Let's go up to 20.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 12, 1, 2,
Okay.
Okay, so it's a striking movie, it's a striking patient. We still don't know why sometimes they can speak, sometimes they cannot speak, why counting or singing. I had a patient that was a singer, she had a stroke, and then after the stroke at 5 o'clock, she was singing opera to her husband, and that was the only communication that they had all day. Basically, she was mute for 9 hours a day, but then at 5 o'clock, she would sing a piece of opera to her husband, and she was still singing. Anyway, after that observation, basically, there has been this plethora of syndromes that were described. So you hit the brain here, you get that deficit, you hit the brain there, you get that deficit. So this is a collection of pretty exoteric neurological syndromes that I know, and I can localize very precisely. And they involve different kinds of cognitive function. They involve introspection, they involve face processing, they involve intention, and so forth. And the whole idea is this observation will be made, and now you have all this very specialized cortical field based on anatomy, the distribution of cell, and the distribution of connection. This gentleman, Corbetta and Brodmann, developed this map, which is still one of the most used map in neuroscience, which is a map of the human brain based on the distribution of cell and connections. And so the idea was, well, okay, if you have a lesion in area 46, now you have a problem with executive control. If you have a lesion in area 7, you have a problem with spatial attention, and so forth. Okay? So basically this idea that the brain is full of experts, and when you hit somewhere, you're going to knock out that particular expertise. Now, we also knew, we've been knowing for a long time, that if you hit the connection between areas, also you have trouble. So this is an old diagram from 1895, from that gentleman there, Lichtmann, who, along with Wernicke, basically described this very simplified model of language. And you see already a diagram. You have nodes and edges, like in a graph kind of architecture. And so the idea is that you can predict the different deficits depending on where you hit, whether you hit a module, a node, or whether you're hitting an edge. Okay, so this is not an old and new idea. This has been around for a long time. And also we know that out of this exercise of correlating anatomy to behavior, a few syndromes, a very few special syndromes are due to disconnection between areas. So according to this model, that if you hit the white matter now, you have this very special problem. So here's some of the syndromes that have been described as related to disconnection in the white matter. a problem with repeating words, a problem with understanding and recognizing objects, a problem with reading, a problem with planning complicated movements with your hand. And what has been observed since then, and this is now, we're now 1980s or so, that when you have a lesion in the white matter, you see those areas here. So these are all regions of structural abnormality in the white matter. And these structural abnormalities are causing cortical dysfunction as measured by, for instance, technique like blood flow or glucose metabolism. You can take a map of the amount of glucose that is utilized by the cortex, and you see that all those regions indicated by the arrows are regions that receive less blood flow, presumably because that lesion is cutting some of the input and the output out of the cortical regions, and so the blood flow is going down. And it has also been observed that as the patient are recovering, basically the blood flow or the metabolism to that piece of brain recovers, presumably through some kind of either rewiring or rebalancing or connectivity. Okay? Now, unfortunately, the story, as any story in science, does not work that well. You know, the more you dig, the more problem you find. So, for instance, in the case of brain, back in 2007, my good friend Nina Dronkers, who was also the person interviewing the Broca case that you saw before, she went to Paris and she asked all the right paperwork, and she was able to scan Broca's brain, okay? For the first time. She told me that she was so scared to drop the brain because this was like, you know, the most precious artifacts of neuroscience. So she was going to put it inside the CAT scan and inside the MRI scan, and finally she was able to scan it. And when she scanned Broca's brain without cutting it, except electronically, you see that the damage is far from being focal, okay? So here's the original lesion, but now you see that all this tissue in the white matter is profoundly altered. All those regions are white, means that the connections are not functioning. So even in the prototypical case of neurology, Broca's case, the connection between location in the brain and behavior is not that precise, okay? The situation got even worse when you start doing thrombolysis. Thrombolysis is a technique by which you're injecting a drug that can basically wash out the clots that form when you have a stroke. Right now we can treat people within 3 hours. Either we give it a drug or we actually go inside the artery and we can pull out the clot. So if you have a stroke, anybody, you have to rush to the hospital because if we can get you within 3 hours, then your chance of recovery is much higher. But when you do now all these scales for stroke, all right, and now you have thousands of patients, and you have a very simplified motor and sensory scale that we use for assessment. When you do a data reduction analysis on this data, thousands of patients, and now you measure what kind of behavioral deficits you find, you do like an ICA or a PCA or a cluster analysis, it does not matter. Basically what you find two clusters that accounts for all the syndromes that people have described in neurology. A cluster for hitting the left brain and a cluster of deficit for hitting the right brain, okay? And basically all the data that you see are basically divided between the gray cluster, which is the left hemisphere cluster, and the white cluster, which is the right hemisphere cluster. So we have a tension here between a very specific modularity and a very gross simplification of behavior, in which now two components are accounting for 85% of the variance of behavioral variability. Now, if you're a psychologist, you're going to raise your hand and you say, well, hold on a second here. This is fine, but you know, this NIH stroke scale is really not specific. You're now measuring memory, you're now measuring attention, you're now measuring executive function. There are many things that you're not measuring, and this is why you see this data reduction. If you were to measure behavior properly, now you would see many more clusters. Maybe not the same as single lesions, but perhaps more clusters than two. So, to summarize this first introductory part, I think what we carry in our pocket as neurologists and neuropsychologists is this idea of local dysfunction due to damage of specialized modules. However, as I mentioned with a couple of examples, the localization of symptoms is not very successful. It's not very precise. In fact, you cannot predict whether someone is going to do well or not, depending on the location of the lesion very well. And finally, most lesions affect multiple brain regions. Therefore, there is a lot of correlation between deficits. So do we need a new framework to think about this? Okay? And I think one framework that has been proposed, of course, is this framework of network-related brain behavior correlations. And the story, of course, is a long story that spans about 25 years, but just to give it in 5 minutes, back in the mid-1980s, positron emission tomography was created. It was a technique to measure blood flow and glucose from the living human brain. For the first time, you can take a subject in the scanner, you can just do something, and you can measure changes in blood flow. This was a revolution. When I first read about this in 1987, I was so excited. I wrote to the people in St. Louis that were inventing this technique, and I said, I want to come and use PET scanning, because at that time I was catching cats. I was doing neurophysiology, and I was sick of catching cats. So I want to kind of catch human subjects. And so that's how I went in 1988 to St. Louis to do those kinds of experiments with PET. Then forward another 15, 20 years, now you have functional MRI. And functional MRI, it's not invasive, you don't have to inject any radiation, and you can measure these changes in circulation and metabolism that are indirectly related to neuronal activity. So if I increase by 30% my neuronal activity, I get about a 5% change in blood flow and about a 25% change in glucose metabolism. And so I can do experiment using functional MRI. And surprisingly, what we found recently, and this is kind of the lecture that I gave when we had the Neuroscience Day, is that the signals in the brain, this functional MRI activity that we measure, is not random, but is organized in networks, okay? So the noise in your brain is not white noise or Poisson noise, is not noise just at all. It's very structured and is organized very specifically in networks, functional networks, as well as structural networks. And all these combination of edges and nodes can be now analyzed using techniques such as graph analysis or mutual information and other things that you know much more than I do. Okay, so this is for the first time we can combine mathematics and physics, statistical physics, and engineering with neuroscience at the whole brain level. And this is really truly a revolution. So based on this, there is a framework that 5 years ago I was interested in testing, which is this idea, can I come up with a new idea, a new way of thinking about brain behavior relationship using these new techniques of network analysis using imaging. And of course I was building on a lot of data coming from neuropsychology and cognitive neuroscience and activation studies for those of you that use functional MRI, suggesting that behavior, of course, is not related to experts in the brain, but is really an orchestra of people, players, interacting together to create a complex behavior. So behavior is supported by distributed large-scale cortical and subcortical networks, not specialized centers, okay? And of course, this is something that is well known and well accepted in neuroscience at this point. The second point is that brain networks, your noise, is constantly exploring patterns that are related to behavior, okay? This is not as intuitive. In other words, your brain noise is not just noise, but it's basically replaying patterns of behavior that are very common. Therefore, by studying changes in brain noise, we might be able to relate that to behavioral deficits, okay? So if your brain noise is altered and that brain noise is behaviorally relevant, then by studying changes in noise, we might be able to understand brain-behavior relationships. And then, this is the other idea, therefore lesions, no matter where they are in the brain, will cause a finite number of abnormality, both in behavior and in neural activity, and these very few canonical components can be described using, for instance, structural imaging or functional imaging. So these are like the canonical ways in which your network breaks down, okay? And of course, since brain networks have evolved to optimize information, and this is true of biological network, of protein networks, amino acids networks, social networks, the internet, of course, then recovery of function should not be due to rewiring at the whole brain level, but should be due possibly to renormalization so going back to the normal state as much as you can is probably the best way to process information, because evolutionary, your brain has developed to segregate an information optimally, segregate and integrate information optimally. So the hypothesis here is that normalization of function, normalization of network function, is the mechanism through which recovery of function after a brain injury happens, okay? So that was the theory. So again, to review kind of a little bit the theory, we started off with this idea that we have experts, vision experts, speech experts, motor experts. Through cognitive neuroscience, we came to this idea of small circuitries in which each element is specialized for a particular operation. Okay, and this is kind of the way we've been thinking cognitive neuroscience. We have language network, we have attention network, we have motor networks. But now, with this network idea, there are no experts. Any node in the brain is basically the influence accumulates and sum the influence of many different inputs and many different outputs. And so specialization is only relevant. And what really matters is the structure of the network. So for instance, if I hit a peripheral network, then the organization of my network will be such that the abnormality will remain focal in the periphery of that brain networks. And one prediction that you might make is that perhaps in that case, structure might matter more than function. In other words, if I'm hitting a local network, the activity is going to stay within the networks, and therefore how much you hit that network structurally, that is going to correlate with behavior. On the other hand, if I go to a node that is connected with many other regions, if I take out that node, now the effect on the network is going to be very widespread, and perhaps the structural damage does not matter that much. What it really matters is the organization and the changes in network function, and that what is going to correlate with behavior. Okay? So this is the hypothesis that we're going to try to test using stroke patients and measuring behavior, measuring structure, and measuring function the best that we can today. All right, so the basic idea is very simple, is we have lots of patients with lots of lesions in different locations in the brain networks. Some of them are subcortical, like in this case, you have a lesion here in the basal ganglia. Some of them are cortical, some of them are in the white matter. Then we get structural connectivity using diffusion MRI. For those of you that know what it is, it's basically a technique to measure the structural connection between brain regions. We also use functional MRI to measure functional connections between the brain. And then we use a lot of behavioral tasks to map behavioral deficits the best we can. And so the three questions that we want to ask in this project is, what are the most common behavioral deficits in stroke? Going back to this idea of many clusters of behavioral deficits versus a few clusters. What is the anatomy of stroke? Surprisingly, in 2016, still we don't know exactly at the population level what is the anatomy of stroke. And then is behavioral recovery of function explained by network dysfunction and normalization, and there is this problem of structure versus function, okay? All right, so here's the study design. So it's a clinical study, but as you see, this clinical study is not just asking clinical questions. This is a clinical study that is asking fundamental neuroscience questions. So, but it's a clinical study, so it requires an interaction between nurses, physicians, radiologists. It's a big deal to put together. So we had money from the NIH for 5 years. We studied about 140 patients, 3 times, within 1 week from a stroke, at 3 months, and then again in 1 year. Okay, all these patients were followed, were seen by doctors, and at each one of these visits, we had a very large behavioral battery, which we measured the best we could in about 3 hours, as many functions as possible. And then we had a very robust imaging assessment in which we look at the lesion location, the structural connectivity, the functional connectivity, and also the perfusion in the brain. Okay? And there are some exclusion criteria. Okay, so the first question is, what are the most common behavioral deficits? And again, the 2 questions is, do focal lesions produce highly heterogeneous behavioral syndromes, or do focal lesions produce a relatively small number of clusters? Okay? And in order to do this, we have all these tests. We perform different kinds of data reduction, cluster analysis, principal component, okay? We try different things, factor analysis. And basically this is work by Lenny Ramsey, who just graduated in the lab. And what we found something very interesting for those of you that are interested in looking at matrices. So basically this matrix that came out not very well. So these are all the tests, and these are all the domains, and the color scale indicates the correlation across subjects in the scores, in the raw scores, okay? And what you can see, first of all, you see that there are clusters domain by domain. In other words, even though I have about 20 tests for language, I have a single cluster of behavioral correlation in language dysfunction. If I have trouble speaking, I also have trouble understanding, I also have trouble reading. If I have problem with memory, I have problem with retrieval of information, I have trouble with encoding of information, I have trouble with familiarity, I have trouble with recognition. These are like keywords, buzzwords for psychologists in the audience, okay? And so forth. So what you see a lot of clustering. And in fact, this principal component showed that a few factors explain a lot of variability. One factor in the motor system explained for 75% of variability across subjects. One factor in memory explained for 67%, language 75%, attention 57%. So, for those of you that are interested in looking at multi-clusters, I see 1, 2, and 3 clusters. And when I look at principal component across domains, now I have 3 scores that are accounting for 70% of variability. In other words, all of my neurology knowledge can be boiled down to 3 scores. And with these 3 scores, I can describe at the population level what happens in my patients, okay? 1 score that is biased toward the left hemisphere, 1 score that is biased towards the right hemisphere. These are the same factors that are seen by the stroke doctors when they're seeing patients. And then they have a cognitive factor that is not measured very well by the NIH stroke scale, okay? And the size of those balls is explaining how much variance you're accounting for. And you see how much variance in numbers, okay? So, now, one might say, okay, well, early on, fine, you find 3 clusters, that's interesting. You know, you don't need to be a neurologist, I just need 3 scores. But maybe at the beginning, there is a lot of problems. There is edema, patients are sick, their blood pressure is through the roof. Maybe there are a lot of non-specific factors that are accounting for this deficit, for this clustering. So the question is, if I follow this patient longitudinally, as the brain is recovering, people are going back home, and I can measure behavior more accurately, do I still see this structure? Is this structure truly related to the brain, or is just some non-specific effects of whatever, having a stroke? And in fact, in this paper that is going to come out soon, we show that these clusters are maintained at 3 months and at 12 months. First of all, we show that there is recovery in these patients, no matter if you have a motor problem, if you have a memory problem, if you have a language problem, you can tell your family that you're going to recover about 70%. There is a rule of so-called proportional recovery. The more deficit you have acutely, the more deficit you will have chronically, and there is a percent, about 70%, no matter from where you start, that's how much you're going to recover. The simple value predicts about 75% of the variability at 12 months. But more importantly, the factor that I described in the same variance, proportional variance, are accounting for behavioral variability at all the 3 time points. Okay, this is suggesting that when you're hitting the brain, behaviorally, your behavioral output is constrained by your network so that you always see the same deficits. People don't change, clusters don't change. You have this very fixed canonical behavioral output after you have a brain lesion in different locations. Because this lesion, as you will see, they're not all in one location. They're kind of fairly heterogeneous. All right, so now let's talk about the second question. What is the anatomy of stroke? And let's think back to Broca and that lesion, very specific lesion, and let's ask this question of modularity and specificity of location. So if you go to Google and you dial stroke images, this is what you see. You see a blood vessel that is occluded, and now you have a cortical infarction. In fact, when people were measuring using CAT scans and X-rays in the past, it turned out that cortical lesions are actually a minority of all strokes, about 15 to 20%. The majority of lesions are in the white matter and in the basal ganglia, okay? If you just do a simple semantic labeling, you just look at the scans and says, yep, cortical, subcortical, basal ganglia, You count them up, and this is what you see. When you do a more modern analysis of this, you take every lesion, and painfully you do segmentation on the lesion, and now you do a probabilistic map of where is the lesion. That's a probabilistic distribution of the lesion in the brain in these 140 subjects. And as you can see, the hotspots are all in the white matter, are deep in the white matter, deep in the basal ganglia. There are very few lesions that are actually hitting the cortex, okay? To some extent, this is biased by the distribution of patients that we have, only 140 subjects. One goal is to extend this analysis to thousands of patients so that we can cover probabilistically the whole brain with enough sampling at each location, but basically the story is not going to change. What is most interesting then when I go and correlate using technique like logistic regression or machine learning, and I try to correlate the behavior of our patients with location in the brain, I see that the more deficits you have, the more you're hitting regions in the white matter and happens to be convergent zones. In other words, areas in the white matter in which you have highway intersection, where there is a lot of traffic in the white matter, if you have a lesion there, now you have a lot of deficits, okay? So these data are demonstrating two things. First of all, that the behavioral correlation that we see is coming out of structural damage in the white matter. And secondly, that this disconnection of the cortex is a fundamental mechanism in stroke in general, not for a few exoteric behavioral syndromes that have been described in the last 150 years. This is the pathogenetic mechanism for stroke. You knock out the white matter, and now you have something going on in the cortex in terms of physiology that must be correlating with your behavioral deficits. All right, I'm going to skip this. So, in this paper that we published last year, the conclusion was that a few behavioral clusters represent the bulk of behavioral variability and should be used as phenotypes for structure function, recovery, genetic, and intervention studies, okay? So with 3 tests, you can classify a lot of behavior. Secondly, white matter damage predicts the severity of impairment and poor recovery, which I'm not showing you. And finally, the white matter damage is common in stroke, hence the secondary physiological disruption of cortical and subcortical network activity must be a major pathogenetic mechanism. And just to kind of leave you a little bit of an iconic memory, single cases, the one that are described by neuropsychologists and neurologists for the last 150 years, are just the tip of the iceberg. The bulk of the iceberg is just this very few canonical component behavioral deficit that I explained, that I described, okay? All right, so let's move on to the third question. Is behavior or recovery of function explained by network dysfunction and normalization of network dysfunction, structure versus function? We can break down the big question in a couple of questions. How activity in cortical network is affected by subcortical and white matter damage, okay? And what is the relative importance of lesion topography versus network activity in predicting different kinds of deficits, okay? And this is work by Josh Siegel, who's an MD PhD. Unfortunately, he's going to graduate in November. And he has done really a terrific job in setting up all these tools and all these analyses to ask these very complicated questions. All right, so a brief blitz on functional connectivity for those of you that are not familiar. Some of you are mechanical engineers or structural engineers. So what I mean by functional connectivity, I mean that I'm measuring the correlation between brain regions in terms of time series, okay? And the signal that I'm measuring is the so-called blood oxygenation level dependent signal, which is basically the amount of oxygen that is present in any particular brain region. And the amount of oxygen fluctuates in the brain depending on the amount of neuronal activity. So for instance, as an example, I take a region in the primary motor cortex, right here. I take a scan for 30 minutes, and I measure the fluctuation of the ball signal in that voxel, in that particular area. Well, you can see that's the red trace there. And then I look at another region in the brain that I know is anatomically connected with the primary motor cortex. And now I take another time series, that's the blue trace, and you see that those two time series are highly correlated. Those regions that are wired together structurally, they also show statistical codependence. there is actually statistically related. You can measure coherence, you can measure correlation, you can cut that, you can do a frequency analysis, there are different things that you can do. You can measure mutual information, does not matter. They're correlated. And when I do a temporal correlation over the whole brain, I highlight a few spots, and those are regions in which the statistical dependency is very strong, okay? And surprisingly, these regions are areas that we know from neuroscience are all connected, and they're all part of the motor system. So the primary motor cortex correlates with this region here that is called the supplementary motor area. It's a region involved in planning your actions. It's also connected with the contralateral motor cortex. It's connected with the thalamus and the putamen, and it's connected with the cerebellum. If I could envision, all these regions are structurally connected. All these regions at rest in the absence of any movement, in the absence of any attempt to move or desire to move. These regions are synchronized together. This is why the noise is not random, but it's highly structured in space and time. When I repeat that experiment over the whole brain, I can divide up the whole brain in networks. And by networks, I mean that the statistical strength of correlation between certain parts of this 45,000 matrix, some clusters show stronger correlation, stronger statistical dependency than others. So within network correlation is stronger than between network correlation. And so all these networks are now projected on the cortical surface of the brain. Surprisingly, they divide up in functional domain that we know makes sense physiologically and anatomically. Just by measuring the brain at rest, your noise partition in ways that make sense anatomically and physiologically. So you have a motor network, you have a language network, you have an attention network, and so forth. This is not trivial. This is very insightful. It's very fundamental. The noise, when you're not doing anything, keep replaying patterns that you use when I actually move. Okay? Now, I can do graph analysis of this, of course, and this network have a certain organization. Some networks are more peripheral, some networks are more central, some networks at a high degree, some networks at a low degree, and so forth. And based on this, I can predict the lesion in different parts of the brain should have different effects. So if I'm eating, for instance, the sensory and the motor networks, they're very strongly connected to the periphery, to nerves, they're connected, they're going inside the brain. It's the first station from the periphery into the brain. If I have lesions there, then those networks perhaps would be more affected in terms of their structure rather than their function, because the functional effect would be remaining within those local networks, okay? It's like you have a computer here in the day, you have a little cluster, okay, you're heating a cluster in an office, okay, the damage is going to stay within the cluster. It's not going to spread to the whole institute. But if you're taking down the server, now the whole day is going to go down. That's the same idea here, right? It's the same as telecommunication. So if I'm meeting now central networks that are involving control, now I'm going to have widespread disruption, and those are the networks that would account for multiple deficits, okay? And in that case, function should be more important than structure. All right, so with those ideas in mind, let's see what happens. So, again, up top, I show you the structure, topography of deficit of stroke, mostly subcortical. In order to do my functional analysis, I use what I call a parcellation. These are basically, I divide up the cerebral cortex in a number of regions. These regions are physiologically motivated based on healthy subjects, okay? So I take a scheme from healthy subjects and I apply it to my patients. Now I have 364 parcels. I have 30 minutes of data. I have a frame resolution of 2 seconds. So I have a lot of data for 140 subjects, and I compute basically all of this to compute differences between my stroke and my healthy subjects, first in terms of mean and variability. And in this paper that was published just a few months ago, we basically show that the overall connectivity structure of the brain is not affected by having a stroke. And this is not surprising, because if I'm meeting the stroke in different locations, the overall topology is not going to change much. So what you see here, you basically see a correlation strength in all these clusters. This is a visual cluster, this is an attention clusters, In healthy subjects, and you can just glance at those two matrices, they're very similar. There is no difference. The spatial correlation is about 0.85. Okay, so they're very similar. However, there are very significant differences. There are actually two phenotypes in the connectivity that are related to stroke. One phenotype is the loss of interhemispheric communication. Okay? So what I'm plotting here, I'm plotting the strength of the correlation in all the connections on the x-axis. Sorry, I'm plotting the cumulative distribution of all the connection by the strength of the connectivity. So you see that controls have a mean correlation of about 0.6 among all the regions. You see that in patients, the correlation is decreased, the curve is shifted to the left, and those are the connection between the left and the right side of the brain, the communication between the two hemispheres. When I look at within the damaged hemisphere, when I look within the undamaged hemisphere, I see that the correlation has not changed much. Okay, this is very interesting. By hitting one side of the brain, I knock out communication between the two sides, but the communication within each hemisphere overall is not very effective. But there is one thing that happens within an hemisphere, which is the segregation between the networks is affected. In other words, the fact that I have segregated modular network in each hemisphere, the fact that I can cluster my data in modules, that property is affected by having a lesion. In other words, the lesion decreases the specificity of network segregation within the damaged hemisphere. So you can see here, for instance, that there is a significant difference between the distribution of strength in this particular graph property between healthy control and patients. I'm going to show you this in a graphic manner, which is more interesting. What is interesting is that these two signals are highly correlated. As I'm losing, if I'm hitting the brain on this side, and I'm losing segregation, at the same time, I'm also losing interhemispheric communication. Okay? So the two signals are actually related, are joint. We don't know which one is driving it. We could do directional analysis like Granger or other kinds of hierarchical analysis to try to understand the directionality of this effect. But right now, we just see a correlation. All right, so now for the question of what matters in terms of behavior, okay? In order to do this, we use a technique called ridge regression. It's a machine learning technique in which basically you're feeding into this model, you're feeding two kinds of properties. You're feeding the location of the lesion. So each brain is basically something like 40,000 location. You have a binary code, damage, non-damage, in each one of these 40,000 points. Then you have 364 voxels by 30 minutes, okay? And all of this is fed as functional information. And what you're trying to predict at the individual subject level is the level of dysfunction for each domain of deficit. So I'm going to try to predict motor deficit, language deficit, attention deficit, and so forth. So just to give you an example, this is a distribution of deficit for motor, okay? And this is actually the distribution of my structural weights predicting my behavior, okay? And this is the distribution of my functional weights predicting my behavior. Now, if you look at the R-squares here, it's about 0.23, and it's about 0.44, which is actually quite a bit of variance accounted for. Structural damage explains very well motor deficits. Functional damage, not so well, okay? And now I can back project the weight of the machine learning onto the brain. So these are the structural weights that are predicting motor deficits. And all those lesions happens to be in motor-related territory, either cortical or subcortical. And those are the functional weights. And you can see here the two phenotypes that I described earlier. I have a disruption of interhemispheric communication, and I have an abnormal increase in the damaged hemisphere in which disconnection are too strong. I have both decrease of correlation and abnormal increase of correlation, okay? So at this point, let's go and look at all the deficits. And what we find confirms the hypothesis that the network organization constrains how well you can predict. So if I'm hitting the periphery of the network, structural damage accounts for more variance than functional damage, okay? So those bars are basically variance accounted for by structural information in white and by functional information in black. And if I'm moving now to cognitive domain, my functional information is more predictive than my structural information. There is one exception, language, in which both structure and function are highly predictive. Okay? So this is basically explaining as both practical implication, what signal you should be looking at for predicting, and also a theoretical implication. How do we think about behavior in relation to lesions? All right, I'm going to skip here some of this because how much time do I have? 10 minutes or 5 minutes or 15 minutes? 15? Okay. So now you might ask, okay, what is the difference then in language or motor or vision or memory? What is different, although the phenotype is the same, what difference is the topology of those connections? So if I'm looking, for instance, at motor deficits, most of the connections that are involved are connections in motor-related networks. If I go into the visual deficits, most of the connections are visually related. If I go into language, it's a much more widespread pattern. If I go to memory, it's even more widespread. Okay? So this is again replaying for you the same idea that local lesions in the peripheral part of the network are causing local functional deficits. Central lesions hitting the server as widespread effect. I'm going to show you in one more way. I can show it by basically highlighting the interaction between the networks. Now I summarize all my parts of network by network. And I'm showing you the joint correlation between network. So for motor, most of the action is in the motor poles, in the motor networks. If I go to vision, most of the action is in the visual network. If I go to language, a lot of auditory action. But if I go to attention, verbal memory, and visual memory, you see that the connectivity between the regions, between the networks is what matters. Okay, there's a lot of information here. But the point is, you can see there is much sparser between network connectivity in those domains and much stronger between network connectivity in these domains. Okay, that's the only thing that I like you to take home. All right, now what about recovery of function? Okay, now I predicted that given the brain has evolved to optimize in this network organization the segregation and the integration of information, rebalancing your network the best you can, going back to normality would be a good strategy. And so we've been using a measure, which is a measure of modularity, which is kind of a nice overall measure of both integration and segregation. And using this matrix of modularity, we'll be looking at recovery of function. Okay? How our networks are changing over time. And what you can see here, in order to do this analysis, you have to threshold these networks at multiple thresholds. Okay? This graph analysis requires basically looking at these metrics at different levels of statistical thresholds. But basically what you can see, you see two functions. You see a function for control subjects, and you see a function for patients at 2 weeks, and you basically see that all the patients have a loss of modularity. Now, when I correlate that particular metric to the functional connectivity abnormality that I described, there is a very strong correlation. So both the interhemispheric loss and the intrahemispheric abnormal correlation correlates very strongly with the modularity. And what is interesting is that as people are recovering, the modularity is returning to normal. Although you have a lesion in the brain, by 3 months, the modularity in your brain is back to normal. By 12 months, it's completely normal. And what is interesting is that the recovery of cognitive function relates to the recovery of modularity. Okay? It's getting a little complicated. I want to just give you one intuitive picture just to show you what I'm describing as modularity. Okay? This is a complicated picture, but we can boil it down to one message. Okay? So up there you see a group of control subjects. These are like 500 control subjects all together, and the strength of that color scale is the standard deviation of the variability, the standard deviation of the connectivity. For each voxel, I measure how variable is the connectivity with all the other regions. So it's a nice global metrics of connectivity strength. Here, I have something called an infomap. It's basically a modularity-driven parcellation, okay? And I divide all my subjects in a particular parcellation that is driven by the metric that I'm using. And this is now is called as a spring embedding techniques, which is basically a way to visualize this relationship. And what you can see here, that these networks are nicely pulled together. And this is what I mean by segregation. So you see, for instance, that this cluster here, which is include the default mode network, is one of the networks in the brain, the most powerful network, and this frontoparietal network are very much segregated by all these other networks that are related in sensory motor control. They're not. When you have now a patient that has a lesion and has a language score that is 12 standard deviation below normal, so this guy is really aphasic, okay? You see that his networks are all bunched up together. They've lost the ability to segregate information. You completely lost the ability to differentiate between what's going on in the default mode network and what's going on in the language network. It's nothing to do with language. All these regions are structurally normal. The patient has a lesion somewhere, but what matters is the organization of his network. As the patient is recovering, and now within a year is going down to 1.2 standard deviation, so he's back to normal, you see that his networks are now pulling apart again. They're becoming again more segregated. Okay, so this is exactly what we're seeing, and we can do this prediction now at the level of single subjects. So in one subject, I can actually look at the trajectory of this, and I can predict how much this subject is going to recover or not based on some of these loss of parameters at the acute stage. All right, so to summarize, loss of intrahemispheric correlation and abnormally high intra-hemispheric correlation are the most robust functional connectivity phenotypes in stroke. Visual and motor deficits are better predicted by lesion topography. Cognitive deficits are better predicted by abnormal network synchrony. And the relative weight of structure versus function in predicting behavior depends on the underlying organization of normal networks. For instance, this distinction between central and peripheral nodes or networks. And finally, the normalization of network synchronization is a preeminent mechanism for recovery. Okay? I want to spend the last 5 minutes, okay, in something that is completely new, but I think there's a lot of potential. I think there's a lot of potential for interaction with physicists and engineers. So, okay, all of this, what I show you right now is correlation, okay? There is no causality. Now, we can go in and try to stimulate these areas using different techniques, either by putting an electrode in the brain or stimulating from the outside, and maybe try to change this network, trying to improve patients. But the question is, what are we treating? Are we treating behavior? Are we treating a connection? What are we treating? And what is the meaning in terms of information processing of these changes in correlation? What does it mean that the correlation between two networks is going up or down? What does it matter? So one way to try to understand this problem is to basically go to modeling of this data, okay, using more principal metrics of information processing in a model. Okay, so one thing that we're trying to do, collaborating with Gustavo Deco in Barcelona. Gustavo is a physicist that has done a lot of statistical physics. And basically, Gustavo, in the last 5 years, he won one of his senior ERC grants, and his work, I was the one introducing him to resting state for the first time, so I take pride into that. Basically, he's been building these computer models in which he takes structural connections from one subject, attaches computational unit to each one of these parcels, okay? So he has a cortical parcellation like the one that I show you, 364 regions, puts a computer node, a node that is generating activity, can be Poisson noise, can be spikes, can be local field potential, can be an oscillation, doesn't matter. And then by the virtue that these nodes are attached through this connection to a biological plausible network, it turns out that now the model replicates at the group level at about 60% what empirically you're measuring with functional MRI. In other words, the model connectivity generated by this computer model is very similar to the empirically measured connectivity. Now, at the group level, At the individual level, nobody's been able to do this yet, mostly because we don't have good structural connectivity model in individual subjects, okay? But this is very interesting, and some of these models have been studied and replicate some of the dynamics of these oscillations. So for instance, we know these oscillations are nested, the model replicates nested oscillations, and so forth, okay? So the idea was, okay, well, what if we damage this network? What if we damage this model, and we try to measure the performance of the model, and then try to relate the performance of the model to the behavior that we observe in the patients, okay? Can we actually simulate a damaged brain? And so this is the idea here, and this is important because, well, I'm going to skip this. This is not relevant. So the basic approach, and this is work by Mohit Adhikari, who was a physicist working between me and Gustavo. So the basic idea is to create a computational model of an injured brain, estimate the processing capacity of the injured model in terms of segregation and integration, and relate the behavior to behavior and functional connectivity empirically model. Okay? So how do we do this? Now we take only patients that have cortical nodes damaged. Okay? Of course, this is important because when you have a white matter damage, we don't really know how the network has been affected. If you have only cortical nodes, now you can remove a node and the relevant connections from the model, and it's kind of a little cleaner. So these are 35 patients. That's a distribution of the lesion, and this patient has a bunch of behavioral deficits, as you might expect. This is entirely pilot data. So then we take our model, we make a lesion, okay? And now we look at how the model of functional connectivity is changed, subject by subject, by hitting the model in different locations. Then we optimize that functional connectivity matrix to approximate, given that some of the connections are not well estimated, we approximate the empirically measured functional connectivity, okay? So we take what we measure in healthy subjects, and we try to approximate the best we can in the model, okay? And then we compute a bunch of different measures. Some of them that can be measured both in the real data and in the model. Some of them can be only measured in the model. And I think that's the beauty of it. So for instance, a model, you can stimulate the model, and you can look at the dynamics of the model. And so by stimulating the model in 100,000 times, you can see what kind of entropy the model can generate, what kind of variability of states the model can generate. And this is something you cannot do in a healthy subject or in a stroke patient because you cannot run the experiment 10,000 times, okay? So this is very interesting because now you can glimpse new information from the model that is not present in the real data. So these are just two metrics, a matrix of integration, which is the largest cluster, and this is a matrix of information of segregation, which is how many states the model generates when you're stimulating a random number of nodes, okay? You stimulate, let it run for 500 milliseconds, you measure the connectivity, you stimulate again, you let it run for 500 milliseconds, you stimulate the topology of the connections by random stimulation. And you do a comprehensive analysis of all the nodes, okay? All right, so what you see here, at the top, you see the structural connectivity of one healthy subject. This is a sparse matrix, as you might expect, based on biological network. That's a damaged connectivity matrix. This is the model functional connectivity in healthy subjects. That's the model functional connectivity in a stroke patients. And this is the optimized, this is the empirical functional connectivity in healthy and strokes. Now, it's not perfect, okay? But there is a very high correlation. If we optimize it at the population level, I can get the model connectivity to have about an 80% similarity at the group level to the empirically measured connectivity. And this is not really surprising. I keep changing and tweaking my weights until I get connectivity that looks kind of what I see in my patients, okay? Now the fun starts, and now I can measure these measures. And what I do here, when I do this exercise, I find that the integration and the segregation measures in the model are decreased as compared to healthy subjects, healthy model, which is not surprising. And I see, and I think this is important, that the number of patterns that I can generate by stimulating the model is consistently lower when you have a brain lesion, okay? Now this is a very interesting observation, that the variability of neural states that your brain can generate is less when you have a lesion in the brain as compared to when you don't have a lesion in the brain. So I think one possible explanation for why the connectivity is abnormal is that abnormal connectivity means lower level of entropy in information states. Now this has to be tested, but one could do an analysis of how much information is coded in stimuli, and the prediction is that a lesion brain should not be able to code as much information when you're, for instance, analyzing stimuli, memories, language, and so forth. The other thing that is interesting is that some of these model parameters relate to the empirical data, but more interestingly, they also relate to behavior. So a measure of information capacity, for instance, across subjects relates to the average attention performance of my subjects. And again, this is very interesting. The more variability you have in neural states, apparently there is very good correlation with how good is your attention. Okay? Again, this is pilot data, but just to give you a glimpse that it is possible now to do a fairly good simulation of a lesion brain and then use some of the simulation data to understand the dynamics and some of the information processing. So in summary, whole brain computational modeling can be used to understand the effect of lesion on information processing. Models are limited in a number of different ways right now. They're limited in terms of the how good are the structural connections. We don't have a good estimate of structural connectivity in single subject. This is why we have to approximate and optimize the model connectivity with the functional connectivity, because that is, we don't have good structural information. You might argue, okay, well, these patterns of stimulation are not really realistic, because now you're just taking random nodes and you're injecting energy in the system without regard to physiological pattern of stimulation. This is something that can be improved, okay? But nonetheless, it is interesting that models can be helpful to simulate stimulation protocols, because now, for instance, you can try to stimulate the brain and ask, okay, what is the pattern of stimulation that is going to get me back the higher entropy? What is the pattern of stimulation that is going to give us back the more variability in neural states? And I bet that parameter, as compared to, for instance, simple functional connectivity, could be a parameter that could be optimized by neurostimulation protocols, such as TMS, electrical stimulation, BCI, and all that. Okay? So, to conclude, I took you through this whirlwind of information, a bit too much. I apologize, but I hope some of the main take-home messages kind of stuck. So we started off with this idea of cortical localization. I show you that neurological deficit post-stroke are not as variable as they appear in single cases or small case series, but there are a few components. This is the behavioral output of a damaged system, is low dimensional. Now, the low dimensionality, and particularly the correlation between deficits, is related to disconnection of the white matter, particularly highways in the white matter, convergent zones in the white matter. And finally, basically that each one of these lesions is causing very specific individualized pattern of disrupted connectivity, and this disrupted connectivity appears to be related to behavioral deficits, accounting for a large amount of variance. Variability explained is very high, and I think this is actually a good way to potentially study, to create what they call individualized medicine, which is a way to basically map in each subject what is the trajectory of the brain over time. After you have a lesion, what is the situation today? What is the situation in 3 months? What is the situation in 12 months? And how can we move the trajectory from an abnormal state of low variability to a higher state of greater variability, using this idea of computational modeling. All right, so to conclude, a lot of thanks to many collaborators and students, particularly Josh Siegel, that has done a lot of the work on prediction, Gordon Schurman, that is with whom I've been collaborating for many years, Lenny, who has done a lot of the behavioral analysis, and Antonello Baldassare, who's actually in Italian from Chieti, who did a lot of work on attentional deficit in these studies. All right, I'll stop there, and I'll take some questions.
The eyes. Very nice talk. It's a fantastic overview. For me, it was really a full experience of joy. Very, very nice and clearly presented. I'm not familiar in the field, but it was fantastic. I have just a very technical question, experimental question. Your population of patients is homogeneous in terms of age, because I don't know if your results can be extended to cases in which the patient can be very young and so with different recovery and so on.
So whenever selecting the sample is very tricky. So the decision that was made 5 years ago was to basically select a clinically relevant sample. So there is no biases. Anybody that comes in with a single lesion that has any kind of neurological deficit, no matter whether it's young or old, that's what we use as our sample. Now, we had to screen something like 6,000 patients to get 150 subjects, 140 subjects that participated. And so there is selection biases, of course. So the most severe patients were not studied. There are several selection biases. But in general, when you compare this sample using kind of epidemiological technique to a larger sample, about 6,000 individuals, which is all the stroke patient in that particular medical center, the population actually is matched for most of the demographics, education, socioeconomic, smoking, diabetes, hypertension, cardiovascular disease, and all that. So it is a clinically relevant sample. Students, we have a rule in neurology now, since I started, that we need to have at least 2 questions from the back. We usually have the residents or the students. It's very important. I see people kind of disappearing in their seats, like, oh, what's going on?
Before they can think of a question, I have one, a more technical one, coming from this department, and it's on algorithms. I mean, there's a lot of data there, and there's a lot of processing there. Yeah. So is there a help that could come from the simplification of algorithms and from new ideas on the algorithmic side? And what kind of improvement can you think on, or relevant improvement on what you can get out of your data from new ideas on algorithms?
Well, there are 2 kinds of, you can split the cake in 2 parts. The first part is the pre-processing of the data. And so a lot of effort goes into cleaning up the data and making sure there is no motion and the quality of the scanning is high, et cetera, et cetera. So some of that is not particularly exciting, I think. The second part, which is all the clustering and all the network analysis, there is a lot to do there, because I just show correlation, which is the simplest possible metric now people are exploring graph. People can look at mutual information. You can do analysis in frequency. All the question of directionality, how directionality is affected by the underlying filter. Partial correlation or methods to partial out variance. All these methods are really being developed right now. So there's a lot to do. And what I show you is just kind of the very, very simple and more pedestrian metric. I think there are also problems of statistical comparison, because none of this is, when you're comparing 150 subjects, 364 by 30 minutes data to 100 by 364 by 30 minutes data, there is a lot of problems of what is the appropriate statistical distribution. It's really not it's not a T distribution, or it's not a Z distribution. So a lot of this is bootstrapping and creating simulated distributions, and what is the appropriate simulated distribution. And of course all the classification is there, all the parts about understanding what are the patterns, what are they representing. All of that is and then all the network modeling part, that is completely open. How you control the network, all this question of controlling these networks, and how you can stimulate different nodes, and how that is changing the network, all of that is wide open. So there is a lot of So I think we had a couple of interesting meetings with people doing telecommunication and system control and all of that. All those ideas about telecommunication can be now, I think, imported into some of this data.
I try to stimulate students' questions because I am in the middle of the road because I am I have a grant, so I am in the middle of the road. I try to stimulate students. And the first question is if I may see one of the first slides where we first introduced graph theory with sensitive and motorial areas for language, even a triangular graph, and I think to understand that for you gray matters represents objects, so nodes, points, and white matter represents line edges, etc. And is it so, or you said also that there are specific areas in white matter which are very sensitive to damage. In your model, that areas are still part of vertices or are edges or are objects, that areas?
No, there are areas in which you have a lot of edges going through.
Okay. And the other one, in your concept of normalization, no, instead of reorganization, is addressed that just to those cases such as strokes where we have lesion, or in other kind of pathologies where we can find alteration in networks such as mental disorders, such as schizophrenia or bipolar disorder, where we can find specific alteration detectable by graph analysis?
My bias is that it doesn't matter whether the pathology, when behavior normalizes, you will go back to kind of this particular arrangement of. So if you have, if you're in depression crisis, I'm sure, if you have like a major depressive episode, and your brain is, for instance, obsessing on particular delusion, and you keep thinking about that, think about depression, right? Thinking about the same thoughts. You use only one filter to filter the whole reality, right? It's a black filter, whatever is coming in is always negative, negative, negative. That's essentially, if you think in terms of, that's essentially one state that is controlling your brain.
No, I was thinking that we have alteration, and the formal networks in another resonance state, even in the euthenic periods of bipolar patients. That was.
I see. Well, I don't know. I mean, I think it's an empirical question, but I think whatever new network you have through reorganization probably is not going to be as efficient as your original network. So although, yes, you may reorganize and you might be able to function, I bet that at some level we can find some deficits. Because, again, this is, it's a history of, it's not just our brain, all the brains are organized like that, from aphasia all the way to, lampreys and zebrafish. I mean, so this is nothing special about the human brain. We should just move away from this idea that we're special. I mean, there's nothing special about our brain. And, they're all organized the same way, the same principle. It's just scaled up with more neurons. But, so I think that organization, whatever that structure is probably the optimal for us.
This is very naive. I know nothing about neurosciences, so please. No. So you mentioned that recovery, you also showed one of the last slides, corresponds also to a regaining of some structure of the network. So I'm thinking about younger people where there is still plasticity in the brain. So in that case, probably there could be recovery without a restructuring, or let's say a return to the original structure of the network. Maybe the brain organizes itself No, I'm just asking, I mean, if that is a possible phenomenon.
Yeah, no, I think kids are potentially an exception to what we talked about. So kids, we know from clinical data that if you have a stroke before age 12, you can actually have a normal IQ. It's kind of amazing if you think about it. So clearly you can remove like completely the left hemisphere, the left cortical sheath, and still you can have normal language or near normal language. So that clearly suggests that there are other solutions that can be found. But I think the key is that the connections are still forming, right? So the network is still forming, and so you can find maybe another solution. But once the network is there, is in place, that particular adult solution. So people have talked about wiring, rewiring locally. This is kind of how people think about plasticity, that you have local rewiring of maps. I think that is small potatoes as compared to what's going on here, frankly. I think this is a much more important signal for recovery. So kids, though, are a very interesting model to study because we know that connection can take, if you have a lesion in the white matter, you see now that the corticospinal tract, rather than going down, it makes these really funny shapes to connect with the spinal cord. So that is something that has not been studied very well.
Yeah. What about the possibility to study the functional properties of neural networks with other techniques, such as oscillatory rhythms with electroencephalography? Can this technique tell us something more about the functional properties, or we don't need it to really understand?
Yeah. Because the resolution of this technique is terrible. It's like one cycle every 10 seconds. And it's terrible because the sampling is slow, but also because the signal is slow. So the signal, if you just hit with like 50 milliseconds pulse the brain, now it takes about 15 seconds for the cycle to complete, right? So what you're seeing here, you see cycle of fast activity that is filtered to a very slow filter. Now EEG, of course, is very interesting, but unfortunately the resolution is not great. I've been doing work with electrocorticogram, electrocorticography, where now you're implanting grids on the surface of the brain. That's better. And MEG, of course, this is the second device that we have to bring to Padua. The magnet, number one, and two, we need to get an MEG system. MEG got better spatial resolution than EEG. But I think EEG and fMRI together actually work pretty well. So a lot can be done with that.
Okay, so thank you for your talk. My question is related to recent research that shows that there are very significant sex differences in terms of patient recovery, especially in language domain. And I know it has been suggested that this is maybe linked to different hemispheric distribution of white matter connectivity among the sexes. So I was wondering whether you have actually observed any sex differences in your sample. Thank you.
If you can send me that paper, I don't know that paper. So we haven't done an analysis I know there is the idea that lateralization of function is different for men and women, and there are maybe some biological substrate of that. I think that whole field is a bit, I think there is a difference at the population level between spatial skills and verbal skills over thousands of individuals. But in terms of the actual neurobiology of that, I think the data, the way I understand it, they're a little unclear, but I don't know this data on recovery functions. So just send me the data and I'll, but we haven't not done an analysis. But we might be able to do that.
Okay. No, this was just in terms of language functions, that maybe females recover faster because they have symmetrical distribution of language network. I think it was a paper in Brain by Forkel from 2015.
Okay, thank you. So before we started, Professor Zampieri told me, yeah, don't worry about this talk, they never went very well anyway. So that was a good reaction. Give me this pulse of stress right there. Got my network dynamics going.
Okay, I think we've done, and we thank again.